Hello everyone, my name is Sahar Bandegi, and today I'm going to talk about human gut microbiome. The gut microbiome refers to the community of microbes living in our intestines. These include bacteria, viruses, and fungi, which play a crucial role in digestion, immune support, and overall health. In this presentation, we will explore this tiny but powerful world and see how it influences our well being. Today, I will explain how the tiny bacteria in your gut, called the microbiome, affect your energy and weight. Scientists studied two diets: a healthy diet called MBD, with lots of fiber and whole foods, and a Western diet, WD, with processed foods. Why? The gut microbiome helps your body use energy from food. Problems here can lead to obesity or diseases. The study asked, does a fiber-rich diet change the gut bacteria and energy balance? What they did. 17 healthy adults tried both diets. MD diet with more fiber, resistant starch like beans, and less processed food. And the WD, less fiber and more processed items. What they found, more energy lost in poop on the MD diet. People lost 116 extra calories daily in their poop. This means the body absorbed fewer calories. And the healthier gut bacteria, MBD, increased good bacteria like Prevotella that make healthy fats. And no hunger or no energy change. People didn't feel hungrier or burn more energy on MBD diets. And why it matters? In the first stage of life, the infant's gut is colonized by a specific set of microbes that lay the foundation for future health. Melanie and colleagues highlight how these early colonizers play a crucial role in not only aiding digestion but also in influencing the immune system and metabolic pathways. The composition of these microbes could affect our health in the long term, setting the stage for disease prevention or susceptibility. The Human Microbiome Project Consortium gave us a groundbreaking view of the diversity within the human microbiome. By mapping the microbes across various body sites, including the gut, they revealed how specialized each community is. And with each microbial group performing unique function that help maintain our health. A balanced microbiome is essential for our well-being. While much was discovered in earlier studies, Leviathan and colleagues expanded this knowledge by uncovering hundreds of new species within the gut microbiome. Understanding of microbial diversity is far from complete, and there is still so much to learn. And as we continue to explore the microbiome, we uncover new species and functions that are essential to human health. Keenan and colleagues took an in-depth look at how diet influences the microbiome, showing the two-way interaction between the host and its gut microbes. Their findings reveal how dietary changes can reshape the microbial landscape and in turn affect energy balance and metabolism. This emphasizes that what we eat not only impact our gut microbes but also how our body processes energy. Finally, Walder and Lee's review brought in an ecological and evolutionary perspective. They explored how changes in lifestyle, diets, and environmental factors have altered the gut microbiome over time. And this research is crucial because it shows how modern behaviors can shape microbial communities and potentially increase our susceptibility to diseases. All of these studies converge to provide a multifaceted understanding of the gut microbiome from the very fast colonizers to the impact of diet and evolution. We now know how important this microbial community is in shaping our health. This research is essential because it seeks to understand how the interaction between our diet and gut microbiome influences our body's energy balance. By studying this relationship, the authors aim to uncover mechanisms that could be targeted to address metabolic disorders like obesity and diabetes, which are increasingly prevalent and place a heavy burden on society and healthcare systems. Specifically, the impact of this research lies in therapeutic insight. By exploring how dietary modification, e.g., a fiber-rich microbiome enhancer diet versus a Western diet, affect the gut microbiome, the study may lead to new strategies that adjust microbial populations to improve energy balance. This could translate into novel, non-invasive intervention for managing weight and metabolic health. Understanding metabolic health. The research addresses fundamental questions about how gut microbes influence energy expenditure, calorie intake, and energy loss, for example, trophical energy loss. This understanding is vital in identifying why some diets may lead to better metabolic outcomes than others. Public health relevance. With obesity and diabetes on the rise, understanding the gut microbiome's role offers a promising avenue to combat these conditions. Think of your gut as a city full of both helpful and harmful bacteria. Then this presentation will answer the following questions. How does the composition of this bacteria shift if we remove fast and processed foods or the Western diet and replace them with more fiber and nutrition options, the embedded diet, and how do such changes help the body absorb fewer calories, for example, by exerting more calories through feces while still not feeling hungrier. Design and methods of this study. In this study, we used a randomized crossover design to examine the individual effects of two diets on energy balance and the gut microbiome. Diets compared. Microbiome enhancing diets focused on fiber resistant starch and minimally processed foods. Western diet contained highly processed, easily digestible foods. Common factors. Both diets provided the same energy and macronutrient composition like fat, protein, carbohydrates. The key difference was in the substrates available for gut microbes. Methods of measurement. Energy expenditure measured using overall calorimetry. Fecal energy measured using COD and PEG for normalization. Microbiome and host responses. Gut microbiome assessment. Whole genome sequencing and 16 rRNA quantitative PCR were used to analyze the microbiome. Short-chain fatty acid (SCFAs). SCFAs were measured in the both feces and serum using chromatography. Host responses, colonic transit time measured using a smart pill device. Interhormones GLP-1 and leptin quantified using enzyme. Linked immunosorbent assay ELISA. Body composition assessed with a DEXA scan to measure weight, fat, and leanness. A statistical analysis, linear mixed models were applied to assess the effect of diet. This study employed a randomized crossover design to compare the effects of a microbiome enhancing diet, high fiber resistant starch, and a Western diet highly processed on host energy balance and gut microbiota. Both diets were energetic and matched for macronutrients, but differed in substrates available for microbial fermentation. Host energy expenditure was quantified via whole room calorimetry, while fecal energy loss was measured using COD and PEG normalization. And by controlling macronutrient identity, this design uniquely isolates the impact of microbial accessible substrates on energy partitioning. Microbiome and host matrix. Gut microbiome composition was analyzed using whole genome sequencing and 16S rRNA, qPCR, short-chain fatty acids, SCFAs, and feces and serum were measured via chromatography. Host responses include clonic transit time, smart pill device, interhormones, GLP-1, leptin via ELISA, and body composition, DEXA scan. And statistical models include linear mixed effects and Pramanova for beta diversity. And this multi-omic approach breaks microbial taxonomy with functional metabolic outputs, revealing diet microbe host crosstalk. Diet-driven microbial shifts. The microbiome enhancing diet increased microbial fermentation, fecal SCFAs, e.g., propionate, and biomass, and diverting energy from host absorption to microbial utilization. Species like Faecalibacterium prevalent in 15 to 19% of samples, and mucin degraders responded inversely to dietary substrates. Bactericides dominated in some subjects, while Firmicute diversified in others, reflecting niche competition. These findings position diet as a lever to reprogram microbial metabolism away from host energy harvest. Host microbe energy dynamics. Microbial biomass account for 25% of fecal energy, with fermentation efficiency linked to interindividual variance in host metabolized energy. On the Western diet, reduced dietary substrate draw microbes to metabolize host mucins, altering niche occupancy. Phylogenetic analysis revealed genomic adaptation in generalysis, e.g. Bacteroides, Tetrautomicron, and Specialis, e.g. Helicobacter pylori. This quantifies the gut microbiome as a metabolic organ, rivaling the liver in energy regulation. Ecological and functional insights. Microbial niche assembly in the gut is shaped by pH, oxygen, and nutrition gradients, with environmental filtering driving diversity. Early life microbiota is influenced by maternal transmission and human milk oligosaccharide, which are selected for symbiotic taxa, and functional redundancy allows microbiota resilience to dietary shifts. Like coral reefs, the gut microbiome forms um, intricate ecosystem where resource competition dictates survival. CFAs and the host physiology. Increased fecal and serum CFAs on the microbiome enhancing diet correlated with um, improved energy, partitioning, and clonic transit time. Propionate alone explained greater than 50% of the variance in host energy absorption, highlighting its metabolic role. Um, CFAs also modulate immune response, as seen in Bacteroides fragilis, mediate T cell priming via PCA polysaccharides. CFAs act as both microbial waste products and host signaling molecules, blurring the line between symbiont and self. Methodological advances. A gut microbiome reference set expanded phylum level diversity by 20% in some taxa, identifying novel species like Faecalibacterium. Genomic analysis revealed niche specific gene enrichment and interspecies interaction. Comparative datasets highlighted population-specific microbial rarity. Each new genome is a puzzle piece in mapping the gut's microbial dark matter. Clinical and evolutionary implications. Novelty, dietary shifts, altered host microbe, competition for a starch, reshaping microbial niche, dysbiosis, susceptibility correlate with bactericides, firmicutes, balance, absence pathogens like salmonella in healthy guts. Um, phage predation further modulates community structure. Human evolution and microbiome evolution are twin narratives. Our genes and microbes co-wrote this story. Limitations and further directions. While this study focused on energy balance, integration, viral and microbiome data could deepen niche understanding. Rural or dysbiotics cohort may reveal untapped microbial diversity. Cross-environmental reference sets are need to dissect host microbe symbiosis. The next frontier, decoding the microbial conversations between bacteria, viruses, and fungi in this ecosystem. Diet modulate the gut microbiome's niche, occupancy, biomass, and SCFA's production, directly impacting host energy harvest. Microbial adaptability and functional redundancy underpin resilience, but Western diets risk mucin degradation and dysbiosis. Maternal dietary and genomic factors collectively um, shape this dynamic ecosystem. In the tug of war between host and microbes for calories, we now have tools to tip the balance toward health. Key findings: diet driven energy dynamics. The microbiome enhancing diet (MBD) increased fecal energy loss, reducing host energy absorption without altering hunger or energy expenditure. Elevated short chain fatty acid (SCFAs), um, linked microbial fermentation to improve energy partitioning. Microbial niche shifts. MBD promoted a fiber fermenting taxa, while the Western diet, the WD, favored mucin degrading bacteria, reflecting substrate-driven niche competition and clinical relevance. Supports fiber-rich diets as non-invasive strategies for weight management and metabolic health. And here are some limitations like a small cohort and short invitation period limit generalizability, um, focus on bacteria and ignoring virion mycobacterium contributions, interindividual variability in microbiome responses not fully addressed, molecular mechanisms or CFA host interactions remain unclear. Further directions like long-term studies in diverse cohorts, um, multi-omics integration to honorable host microbe crosstalk, explore probiotics, prebiotics to amplify NVD effects, investigate lost microbial diversity in modern diets using rural traditional populations, develop personalized nutrition models based on baseline microbiome profiles. For conclusion, harnessing the gut microbiome, how a fiber-rich diet modulates energy balance and combats obesity through fecal energy loss. Fiber driven microbial shifts, enhancing fecal energy loss and metabolic health via SCFA production, microbiome targeted nutrition, and non-invasive strategy for weight management through enhanced fecal energy loss. and gut microbes as calorie regulators, and the role of dietary fiber in reducing host energy absorption, and dietary fiber and gut microbe synergy, reducing host energy absorption to tackle obesity and metabolic disorders. This study showed that a fiber-dense microbiome total diet MBD increased substantially fecal energy loss by shifting gut microbial composition toward fiber fermenters, e.g. Pervodella, and increase in short chain fatty acid (CFAs) production, thus reducing host energy absorption without impacting hunger or energy expenditure. Uh, the results squarely address the research questions. Dietary fiber enhances fecal calorie excretion by diverting energy away for microbial utilization. MBD promotes beneficial bacteria and SCFA's production when compared with the Western diet, and increased fiber intake helps in weight management by blocking the absorption of calories instead of suppressing appetite. This work highlights the important role of gut microbiota in energy management and offers a non-invasive diet-based approach to tackle obesity and metabolic disorders. The limitation include small sample size and short intervention periods, but will necessitate further investigation, and the work is a step forward in understanding diet, microbiome host interaction, and emphasizes the role of personalized microflora targeted nutrition in promoting public health. And here in this slide, I have couple of questions for group discussions, and in this slide, I put my references. Thank you for your attention and interest.